Hello, my name is Julia Garrow, and here is my name and date. I apologize for any background sound or commotion. I'm at work right now recording this. My presentation is on chemoreception, smell, and taste. I apologize for the formatting of my presentation. I couldn't figure out how to use the slideshow and the record thing at the same time, so this is what we're working with. Olfactory is one of the oldest senses. It was one of the first senses discovered by scientists. The pathway, which we'll get into more detail in the next slide, consists of olfactory epithelium, which lines your nasal cavity, sensory neurons, cranial nerves, the olfactory bulb, tract, and olfactory cortex. And like I said, we'll get into more detail on the next slide with that. Scientists have discovered that smell, memory, and emotion are very closely linked. A lot of times people can smell a cologne or a certain scent, and then it reminds them of something that happened in the past or makes them feel a certain way. Another thing that we'll get into more detail in the next slide is that olfactory receptors are G protein-linked membrane receptors. So now we'll get into more detail regarding the olfactory pathway and olfactory transduction. The first thing that needs to occur in order for anything to be smelled is that the odor molecules must dissolve in the mucus first. The mucus is secreted by the olfactory epithelium, lining the nasal cavity. The epithelium is embedded with primary sensory neurons that are called olfactory neurons. The axons of the olfactory neurons they form the olfactory nerve, which is also called a cranial nerve. These nerves synapse with secondary neurons in the olfactory bulb, which is located on the underside of the frontal lobe in the brain. And secondary or higher neurons then project through the olfactory tract to the olfactory cortex. And from the cortex, they can either travel to the cerebral cortex or the limbic system, where they can connect with the amygdala or hippocampus, which is where this emotion and memories get linked. And I mentioned in the last slide that olfactory reception has G protein linked receptors, and odorant molecules activate the special G protein called GULF. And when this protein is activated, it increases intracellular CAMP, and this increase in the CAMP opens the CAMP gated cation channels, which depolarizes the cell. And we know that if the depolarization is strong enough, then that triggers an action potential, and then the odor can be sensed. The sense of taste, or gustation, which is also referred to, is essentially made up of 5 different sensations. There's sweet, sour, bitter, umami, and salty. Umami is triggered by the presence of glutamate. All of these are triggered by different things in the cell. So sour is triggered by hydrogen ions, salty is triggered by sodium ions, and so on. One of the things that scientists have discovered is that a bitter taste is typically a warning of toxicity. So when you eat something bitter, you are automatically inclined to try and spit it out. The sweet and umami tastes are usually associated with nutritious foods like fruit, things like that. The gustatory pathway is a little bit more complex than the smelling pathway, but it is very similar in a lot of ways. First, we have taste buds that are composed of 50 to 100 taste receptor cells, along with support and regenerative basal cells. Similar to smell, the tastant must be dissolved in the saliva first, in the mouth, and the general pathway includes the apical membrane protein, gustatory neurons, cranial nerves 7, 9, and 10, the medulla, thalamus, and insula. Signals from taste and from the neurons from tasting things triggers digestive system activation, which will basically tell us that we need to keep eating or stop eating. Basically, the 4 different types of taste cells are very sensitive to different things. They usually sense each of the tastes that we talked about before. Some of them sense up to 3 of the sensations that we talked about previously. Though the transduction has differences within it, they follow the same general pathway, which is that the ligands, or the tastant, activates the taste cell once it is dissolved in the saliva. And then this is where things get different. So there are intracellular pathways that are different, and we can see here that sweet, bitter, and umami taste have G protein coupled receptors, whereas the salty and sour taste have ion channels, and that's kind of the difference between the two. Then the calcium signal in the cytoplasm triggers exocytosis or ATP formation, and then the neurotransmitter or ATP is released, and the primary sensory neuron fires and active potentials are sent to the brain. And that is the general overview of the gustatory transduction. It gets into a lot more detail and a lot more complex than the smell transduction, but it follows the same general principle. Each type of the taste receptor cells have different types of names. So the type 1 cells are support cells, and they are sensitive to salty flavors. The type 2 cells are receptor cells, and they are sensitive to sweet, umami, and bitter. Type 3 cells are presynaptic cells, and they are sensitive to sour taste. And then type 4 is basal cells, and scientists are kind of still figuring out what the basal cells are sensitive to and their function within the transduction pathway. So at the moment, they are kind of just support cells. The details of the complex gustatory transduction pathway is still being figured out by scientists. It is largely unknown with the nitty-gritty, which is why we have this general outline, and it's basically the similar pathway to the smell transduction, but this is pretty much as much into detail as we know this far. We know that the type 1 cells that sense the salty, the type 3 cells that are sensitive to sour taste, we know that when the hydrogen ions enter the cell through the channels is when it is able to bind to the receptor, whereas the sweet, umami, and bitter ligand kind of just bind to the G protein coupled receptors as I listed up here. Salty is also kind of sensed similarly to sour tastes through the ion channels. This is the study tool I created, which is basically a treasure map where you follow through the smell and taste transduction pathways. Both start with the dissolving in mucus, and then you follow each step through the map until you get to the end. As you can see here, the smell pathway ends with 2 different pathways, as I had mentioned before, that the signal can go to the cerebral cortex, or it can follow the pathway to the limbic system, which is where memory and emotion can be triggered from the scent. Taste ends at one location, which is the gustatory cortex in the insula, located in the insula in the brain, after it passes through the thalamus. I think this visual helps to clear up the pathway, especially the order of the pathway. I think it's easy to read, it's kind of fun, there's fun colors, and I think it would help a lot of people learn the pathways and the order of which the pathways are followed. This is the citation I used. Where I got all my information from is just from Silverthorne, the book. Thank you for listening to my presentation.